We will learn about antigen-antibody reaction. So let's begin. Today we will learn about antigen-antibody reactions. Antigen-antibody interactions underlie many immunological techniques in which the high specificity of the antibody is used to identify, isolate, or quantify a particular antigen. Immunology has, however, developed a number of its own technique, particularly those based on the antigen antibody interaction. For example, any molecule that act as antigen can be identified in tissues by immunocytochemical methods. Very low concentrations of such molecules can be quantified by radioimmunoassay, in short RIA, and enzyme-linked immunosorbent assay, it's in short ELISA, ELISA. There are hundreds of different immunological methods now being used. Some of the most common methods are we will now discuss. Now, first is precipitation reaction. First point is under this we will study about classic precipitin reaction. One of the first observation of antigen antibody reaction was their ability to precipitate combined when combined in proportion at or near equivalence. This is seen in the classic precipitin reaction where antigen and antibody are mixed in solution, and as increasing concentrations of antigen are added to a constant amount of antibody, the amount of immune complex precipitin rises and then falls. The precipitin curve generated in this way has three zones. First is antibody excess zone, equivalence zone. We will study onwards. Okay. So let's begin with antibody excess zone. The amount of antigen is insufficient to react with the precipitate all the antibody present. Thus, free antibody can be detected in the supernatant. Next point is equivalence zone. The added antigen is sufficient to combine with the precipitate all the antibody present, and neither free antigen nor antibody can be detected in the supernatant. Third point is antigen excess zone. The amount of antigen exceeds that required to bind all the antibody, and this leads to a reduction in the amount of antibody precipitated. This fall is due to the formation of soluble antigen antibody complexes by the excess antigen. The extent to which this phenomenon occurs varies with different antibodies and with the species from which the antibody is derived. Now let's study about precipitin reactions in gels immunodouble diffusion. By performing precipitin reactions in agar gels, it is possible to distinguish separate antigen-antibody reactions produced by different populations of antibody which are present in a serum, the immunodouble diffusion technique. This technique has been extended to the examination of the relationship between different antigens. In this process, agar gels are poured into slides and allowed to set. Wells are then punched in the gel, and the test solution of antigen and antibody are added. The solution diffuse out, and where antigen and antibody meet, they bind to each other, cross-link and precipitate, leaving the line of precipitation. The precipitin bands can be visualized by washing the gel to remove soluble proteins and then staining the precipitin Rs with a protein stain such as coma C blue. This technique may be used to determine the relationship between antigens and a particular test antibody. Three basic patterns appear. First, in reaction, the precipitin arc formed between the antibody and the two test antigens fuse, indicating that the antibody is precipitating identical epitopes in each preparation. This does not mean that the antigens are necessarily identical. They are only identical in as far as the antibody cannot distinguish a difference. The reaction, the antibody preparation, distinguishes the three different antigens which form dependent precipitin areas. In reaction 3, the antigens share epitope 1, but one antigen, this has the antibody can distinguish them by virtue of being able to react against both epitopes. A line of identity forms epitope 2 has reacted secondary with the second epitope, thus indicating partial rather than total identity between the antigen preparations. Now let's study about the immunoelectrophoresis. Some antigen mixtures, however, are too complex to be resolved by simple diffusion and precipitation, and so the technique of immunoelectrophoresis was developed. Antigens are separated on the basis of their charge before being visualized by precipitation. Immunoelectrophoresis allows the comparison of complicated mixtures of antigens such as are found in serum. Antigens are separated in an agar gel by placing an electric charge across it. The gel's pH is chosen so that positively charged proteins move to the negative electrode and negatively charged proteins to the positive. A trough is then cut between the wells and filled with the antibody which is left to diffuse. The antigens and antibody form precipitin arcs. The gels techniques only identify antigens and antibody qualitatively, but by further modification using the technique of single radial immunodiffusion, they can be made quantitative. Now let's study about single radial immunodiffusion. Single radial immune diffusion allows quantitation of antigens. Antibody is added to the agar gel which is then poured onto slides and allowed to set. Wells are punched in the agar and standard volumes of test antigen to different concentration are put in the wells. The plates are left for at least 24 hours, during which time the antigen diffuses out of the wells to form double complexes with the antibody. These continue to diffuse or outwards, binding more antibody until an equivalence point is reached and complexes precipitate in a ring. The area within the precipitin ring, measured as ring diameter squared, is proportional to the antigen concentration. Unknowns are derived by interpolation from the standard curve. The whole process may be reversed using an antigen containing gel to determine unknown concentrations of antibody. By applying a voltage across the gels to move the antigens and antibodies together, immunodiffusion becomes counter current electrophoresis and single radial immunodiffusion becomes rocket electrophoresis. Now let's study about the countercurrent electrophoresis. Countercurrent electrophoresis is performed in agar gels where pH is chosen so that the antibody is positively charged and the antigen being tested in is negatively charged. By applying a voltage across the gel, the antigen and antibody move towards each other and precipitate. The principle is the same as for immuno-double diffusion, but the sensitivity is increased to 10 to 20 fold. Now let's study about the rocket electrophoresis. Antigens may be quantitated by electrophoresing them into an antibody containing gels in the technique termed rocket electrophoresis. The pH of the gel is chosen so that the antibodies are immobile and the antigen is negatively charged. Precipitin rockets form. The height of the rocket is proportional to antigen concentration and unknown are determined by interpolation from standards. The appearance of stained rockets antibody having different charges rockets is shown on the right. Both techniques rely on the antigen and antibody having different charges at the selected pH. This is true for most antigens since antibodies have a relatively high isoelectric point. If the charges on the antigen and antibody do not differ sufficiently, the antibody or antigen can be chemically modified to alter its isoelectric point. Rocket electrophoresis can be reversed to estimate antibody concentration if a suitable pH gel can be found to immobilize the antigen without damaging it to preventing the antigen antibody reaction. Now let's study about agglutination reaction. All antibodies have at least two antigen binding sites and most antigens have at least two antigenic determinants. Thus, antibodies can cross-link antigens producing aggregate termed immune complexes. Immune complexes are sometimes designated according to their physical properties. For example, when an immune complex containing a soluble antigen becomes large enough to precipitate, it is called the precipitin. When a complex is formed by cross linking cells or other particles, it is called an agglutinin. The reaction is called agglutination. Agglutination reaction can often be detected with the unaided eye when microscopic particles aggregate into large clumps. In the first agglutination reactions studied, blood cells or bacteria were employed as the particles. These are naturally coated with antigens that can be cross-linked. Recently, techniques have been developed to coat microscopic latex spheres with antigens, providing synthetic particles useful in agglutination reaction. The best known use of agglutination reaction is the typing of human blood, a technique discovered in 1900 by Karl Landsteiner. He demonstrated that four types of human blood A, B, and O can be distinguished by simple agglutination test. For example, blood from a type B individual is clumped by serum from a type A individual but not by serum from a type B individuals. Before blood typing made it possible to match the blood of donor with that of a patient, transfusion were rarely performed because of fatal reactions that often occurred when untyped blood was used. The example of a diagnostic test based on the agglutination of microscopic latex spheres is one of the modern pregnancy test. It detects the greatly elevated concentration of the hormone human chorionic gonadotropin, in short HCG, that occurs in urine and blood early in pregnancy. First, a simple, usually urine and blood early in pregnancy. First, a simple urine from the patient is mixed with a solution of antibody specific for HCG. In the second step, latex spheres coated with HCG are added. If present at high enough concentration in the first step, HCG will bind to most HCG specific antibodies, thereby preventing them from agglutinating the latex pairs. Now let's know about the neutralization reaction. Let's know about the neutralization neutralizing reaction. One of the simple effector system antibodies can combat certain pathogens just by binding to them. Immunity to diseases like tetanus and diphtheria, in which pathogenesis is largely the consequence of the action of a toxin, depends on antibodies in blood that bind to the toxin and inactivate it, a process ter- a process termed toxin neutralization. The resulting antibody toxin complexes cannot absorb to toxin receptors on host cells. Similarly, antibodies bound to the surface of virus prevent adsorption, and such antibody mediated inactivation is termed virus neutralization. For example, antibody to the outer coat proteins of some rhinoviruses which cause colds can prevent the viral particles from binding to and infecting host cells. Now let's study about the complement and lytic reaction. The complement system is a group of about 20 serum protein whose overall function is the control of inflammation. The component interact with each other and with other element of the immune system. Synthesized by hepatocytes and monocytes, these proteins help, that means complement antibody response responses and have a wide spectrum of activities including a pivotal role in innate diffuse mechanism. For example, a number of microorganisms spontaneously activate the complement system via the so-called alternative pathway, which is an innate non-specific reaction. This results in the microorganism being coated by complement molecules leading to its uptake by phagocytes. The complement system can also be activated by antibodies bound to the pathogen surface via the classical pathway. When it co-mediates a specific adaptive response, and complement activation is a cascade reaction with each component sequentially acting on others in a similar way to the blood clotting system. The complement system lyses microorganisms and infected cells by forming holes in their plasma membrane. Now let's study about the complement system. The complement molecules include C1, that is C1Q, C1R, C1S, C2, then C2, C3, C4, C5, C6, C7, C8, C9. As well as C means complement system. Okay. As well as a set of molecules which are primarily associated with the alternative pathway, including factor B and factor D. C3 is the pivotal molecule of the complement system. Two principal pathways are involved in complement activation. All of which coverage on the activation of the third component, that is C3. These are, first is the classical pathway. Let's know about the classical pathway. The classical pathway, which is activated by antibody bound to antigen, and two is alternative pathway, that is alternative pathway activated in the presence of various microbial pathogens. The proteins of the system act in enzyme cascades, where each step generates enzymes which act in some steps of the cascade. Thus, a smell initially stimulus can rapidly generate a large effect. Both classical and alternative pathways generate a C3 convertase which converts C3 to C3b, the central event of the complement pathway. C3b in turn activates the terminal lytic sequence C5 and C9. The first stage in the classical pathway is the binding of antigen to antibody. The alternative pathway does not require antibody and is initiated by the covalent binding of C3b to hydroxyl and amine groups on the surface of various microorganisms. The alternative pathway provides non-specific innate immunity, whereas the classical pathway represents a more recently evolved link to the adaptive immune system. Now let's study about the classical complement pathway. Blood contains a group of 9 proteins that is designated C1 through C9, collectively termed complement that function in a complex series of reactions to kill some kinds of foreign cells. The first reaction sequence to be demonstrated, termed as the classic pathway, is triggered by the binding of single IgM molecule of Ig1, Ig2 and or Ig3 to an antigen molecule, resulting in a conformational shift in antibody structure that exposes a receptor for C1 on the CH2 domain. C1 is composed of three subunits C1Q, C1R and C1S. C1Q is a hexamer of three kinds of chains A, B and C, each about 23 kilodalton. Kd means kilodalton. The high proline content of C1Q suggested that it has an unusual structure. Indeed, each ABC trimer forms a triple stranded helix of the kind seen in collagen. The overall structure of C1Q resembles a bunch of six tulip. Each of the six globular heads contains a binding site for the Fc moiety of an antibody molecule. C1 is not activated by an antibody molecule alone or by a single one complexed with an antigen. Rather, a group of several Fc units serves as the trigger when they are brought together by the formation of a multivalent antibody antigen complex. C1R and C1S are located in a cavity formed by the heads and stems of C1Q and the simultaneous binding of several Fc units to the globular heads of C1Q converts C1R from a zymogen into a protease. This activation step in which C1R undergoes an internal cleavage is reminiscent of the activation of pepsinogen by acid. It seems likely that the binding of Fc units pepsinogen by acid. It seems likely that the binding of Fc units to C1Q pulls an inhibitory segment out from C1R. Activated C1R in turn splices a peptide bond in C1S to convert it into an active protease. Binding of C1Q subunit to adjacent complement receptors results in a conformational change of the C1 complex. As a consequence, C1S becomes proteolytic active and cleaves C2 into fragments C2A and C2B. and also clips C4 into fragments C4A and C4B. C4b binds to antigen antibody C1 complexes. The resulting AgAbC1C4b complex is stable. But binding activity of a free C4b is quickly lost. C2A adsorbs a bound C4b to form a proteolytically active complex that cleaves C4AC 2b, forming the complex C4bc 2a C3b that cleaves C5 into fragments C5a and C5b. C6 and C7 rapidly bind to C5b, forming a C5bc6C7 complex that possesses an unstable membrane binding site. But once bound to the membrane, this complex is stable. Then C8 and C9 bind, forming the complex C5bc 6C 7C 8C 9 membrane attack complex, that is MAC, that creates a pore in the target cell membrane. If the cell is eukaryotic, its cytoplasmic contents rapidly leak through the pore, causing death of the target cell. These pores have diameters of about 10 nm and permit leakage of intracellular components and influence influx of water, which result in disintegration of the cell. If the cell is a gram-negative bacterium, lysozyme in blood enters pores made in the outer membrane and digests. The gram-positive bacteria are resistant to the cytolytic action of complement because they lack an external membrane. Now let's study about the alternate complement pathway. In the absence of antibody that binds to gram-negative bacteria, complement is still bactericidal. This is the consequence of the alternate complement pathway that brings with cleavage of C3 into fragments C3A and C3B by an enzyme normally present in blood. These fragments are produced at a slow rate and usually do not trigger the next step, the cleavage of C5, because free C3b is rapidly broken down into inactivated fragments. However, C3b is stabilized by binding to lipopolysaccharides of bacterial outer membranes or to IgA and IgG antibodies in immune complexes. A protein in blood called factor B adsorbs to bound C3b, leading to formation of an active enzyme denoted C3b capital B small b, that is further stabilized by a second blood protein properidine, resulting in efficient formation of C5b from the cleavage of C5. The steps that follow in the alternate pathway are identical to the terminal steps of the classical pathway. Now let's study about the functions of complement system. The principal functions of the complement system we will study now. First one is opsonization. Complement components coat the bacterial surface, allowing the phagocytes to recognize the bacterial and engulf them. This process is called opsonization, but neutrophils and macrophages possess on their surfaces a receptor for C3b. Therefore, C3b that is bound to the surface of a pathogen can form a breeze that facilitates phagocytosis. Indeed, opsonization appears to be the most important function of complement. People who lack C3 are usually much more susceptible to bacterial infections than are people who lack some other complement protein. Now let's know about chemotaxis. Polymorphs and macrophages have specific receptors for small complement fragments that are generated during complement activation. The Fragments diffuse away from the site of activation and stimulate chemotaxis. For example, complement fragment C5a, which is a very potent chemotactic factor, causes macrophages, neutrophils, eosinophils, and basophils to concentrate at the site of complement fixation. Now let's study about the lysis of target cells. The final step is complement activation causes the assembly of a membrane attack complex in short MAC, which are inert itself into lipid bilayers such as the outer membrane of gram negative bacteria or a viral envelope. If the complement system attacks mammalian cells, example the erythrocytes from an incompatible blood transfusion, then osmotic disruption of the target cell ensures causing lysis. Fourth point is increased blood flow to the site of activation and increased permeability of capillaries to plasma molecules. Fifth point is release of further inflammatory mediators from mast cells. Now let's study about the phagocytosis. Phagocytocytes arrive at a site of inflammation by chemotaxis. They may then attach to microorganisms by way of their non-specific cell surface receptors. Pathogens such as bacteria are taken off by binding to opsonic receptors including the Fc receptor, complement receptor and the receptors for carbohydrate, in short MFR. The second point is the particle is engulfed and phagosome forms. Third point is acidification of the phagosome follows as toxic molecules, reactive oxygen, RO, and nitrogen intermediates are pumped into the phagosome. The marked Fa by 11 is located in the phagosome membrane. Fourth point is lysosomes fuse with the phagosome releasing proteolytic enzymes into the phagolysosome, which digest the bacteria. Undigested microbial products may be released to the outside. Next point is on occasion the membrane of the phagolysosome is disrupted. Antigenic fragments may then be bind MSC class T molecules and become diverted to the acidic endosome compartment for interaction with MSC class 2 molecules and antigen presentation. The process induces secretion of toxic molecules and cytokines. Finally, antigen antibody reaction finished. Any questions regarding this, you can ask me in the comment box and subscribe my channel to know more updates. Thank you.